Welcome to New York Bio's virtual breakfast series. A digital program started in 2020 bringing you fireside chats with leaders across the healthcare spectrum. This week's episode features Stacy Blain, a co-founder and the CEO of Concarlo Therapeutics, and Arthur Klausner, the executive chairman of Concarlo Therapeutics.
Hi, everyone. Happy Tuesday. My name is Jennifer Hawkes-Bland, and I'm the CEO at NewYorkBIO. We are very excited that you're joining us for another edition of our virtual breakfast series. As always, I almost always forget the housekeeping and then remember it at some point during my introduction. We know that you might have questions for Arthur and Stacy today as we talk about Concarlo and their development and the New York ecosystem. So please ask those questions in the chat or the Q&A, and Derek and I will get to them throughout the course of our conversation. One other upcoming New York Bio event that I definitely want to put on your radar on Sunday, February 5th. I probably have that date wrong. Whatever the Sunday is.
You got it.
Yeah, okay. Sunday, February 5th. We are hosting a welcome reception in Midtown. Sorry, it's in Times Square, but that is where the Bio CEO conference starts the next day. So we want it to be walking distance for people that are flying in from out of town. So come join us for a reception that evening. The registration is on our website. So if you're in town that Sunday evening, please join us. Good news is it is not Super Bowl Sunday this year because that was going to be difficult last year, but luckily COVID had us push it so everyone could also enjoy their Super Bowl parties, which might have also been super spreaders, but we won't go there this morning. So we are very excited for our conversation today. So I want to hop right in it before we waste any more time. Over to Derek to kick off the conversation. Thanks, everybody.
All right. Good morning, Stacy and Arthur. As I said on Twitter the other day, the only thing that I think is better than having one terrific New York City entrepreneur on this program is having two of them. So Arthur, I've known for quite a long time and always had an immense amount of respect for, and Stacy for the last few years. I have had people simply tell me how wonderful you've been, and I have no doubt if we weren't in the midst of a global pandemic that we would have met far earlier than this. As it stands, I am super excited to have both of you here. Concarlo is a great New York City story, and I'm excited to dig into it. So why don't we kick off? So as you guys know, we like to start with a bit of an origin story. And Stacy, as the founder, why don't we start with you?
Thanks for having us here today. Very excited to sort of tell the Concarlo tale. So this Concarlo really is a New York City story. I am educated in New York City, did my undergraduate at Princeton, and then I got my PhD at Columbia, and then I moved to Sloan Kettering, and then I started my own laboratory at one of the SUNYs. So really haven't left this tri-state area since I decided to become a scientist. And really am a cell molecular biologist. I had always wanted to be that. I'm from Florida. I moved to the Northeast to study molecular biology at Princeton and then just never left. And always was very focused on a target. I did my graduate work with the great Steve Goff, and we studied HIV and retroviruses, and we studied in particular reverse transcriptase. So it was really focused on learning about a particular protein as a modality to understand biology, but also with the sort of the itch in your head of this could become a therapeutic target, which of course reverse transcriptase did and has been. And so started at Sloan Kettering. I worked with Juan Massaguet, another great New York City great. He runs the SKI Institute. And he and Andy Koff had just discovered this protein P27, which at the time we thought was just strictly an inhibitor of CDK4 and CDK6 and CDK2. And I was really the first person in the door to work on it and, really have spent the rest of my career, about 25 years, just focused on that protein and thinking always from the idea of I want to understand how this protein, where is it in the cell? Who's it talking to? What makes it motivated to be on, motivated to be off? Thinking about it very protein centrically. And then also with the idea that if we understood how this protein worked, we could harness it, we could leverage its potential as a therapeutic target. And so that was really in the back of my mind as early as my postdoctoral days. And, we sort of mused about the potential of this. And so I started my own laboratory, and I just dug in, writing grants and teaching students and being in the classroom, but also doing really interesting work to ask those questions. And then we essentially discovered how P27 turned these three main kinases, CDK4, 6, and 2, on and off in the teens. We patented the technology, and then I started Concarlo in 2017. And so we're sitting here now just a few years later. And essentially, when I started it, we can get into the details of sort of the serendipity about the starting of that company and the factors that caused me to start it. But, it was really an academic idea. It was an idea we had on a piece of paper. Look, this is a great target. And if we could do this, this would be amazing. And so now we are here in 2023 with a drug that actually we finished developmental manufacturing of this drug, and we're about 18 months from our first in-human clinical trial for drug-resistant metastatic breast cancer. So it really is a long journey for me, in the sense that this was what I was thinking about, over 25 years ago, but it's also been a journey that has unfolded at every step of the way with new insights and new opportunities. So, very New York City based, and very happy to have been able to take this from academia into biotech.
That's awesome. Okay, I have a really good question that I'm going to park for a couple minutes while Arthur can give us his background. But it's basically going to be germane to how did you actually get to, how did the idea get to an actual company? I'm going to park it for a sec. Arthur, why don't you go and let us know where you came from too, because I think everyone's going to realize that this is a pretty fantastic combination of two people here.
Yeah, thanks, Derek and Jennifer. Really appreciate the opportunity to be here. I was thinking about this, and my only regret is that I'm not going to be able to have my Tuesday morning business guilty pleasure of watching this. But I appreciate what you guys have done, and congratulations on what you've been able to accomplish through the pandemic and post-pandemic. So again, thank you.
Arthur, I've been called many things, but guilty pleasure is not one of them.
That's a nice one.
I was like, I have to write that down.
For the record, I was staring
guilty. Does that count?
I was staring longingly at Derek when I said it. So it's
It doesn't make it much better, but
no, honestly, Arthur, make better choices with your life.
On that note, but I guess my New York story, and at the risk of Derek rolling his eyes, began when I was a little kid growing up on the Upper West Side. And when it would rain on a afternoon or a weekend, my mom would drag myself and my little brother to the American Museum of Natural History. And I kind of thought if we'd grown up on the Upper East Side and she was taking us to the Metropolitan Museum of Art, I probably wouldn't have gotten into the whole biology thing. But that's really where it started for me. I ended up majoring in biology in college, and joined right after college a startup that was a publication, what's now Nature Biotechnology magazine. And that was an interesting era. So this is 1982. Was able to do a bunch of things at that startup, including, I believe, putting together the first ever biotech stock index. And I remember back then, it was a monthly publication. At the end of the month, I would get out the Wall Street Journal. I would write down the closing prices. I would take out my calculator because I didn't know how to use VisiCalc. And I would compute the numbers. So that sort of takes you back to where that was. Got really interested in the interface between science and business, went off to business school. And in 1990, on graduation, I joined Domain Associates, which was one of the first life science focused venture capital funds. At that time, we were the largest such fund with our $41 million fund. So that goes to show how things have changed. We grew to raising $500 million funds. I moved over to Pappas Ventures, that was a little bit smaller in scale. And from there, I moved on to the other side. So it feels like a random walk to me, but when I think about it as a writer, I was pretty far away from the industry, sort of a voyeur reporting on it. As an investor, a little bit closer, and then as an entrepreneur and as a manager, a lot closer. Not as close as the doctors, of course, and they have my undying respect for what they do, but kind of closer still. So after Pappas, I did 3 startups, an oncology company, an ophthalmology company that were both sold, and then a spin out from Sloan Kettering in the nephrology space. In the meantime, I had met Stacy. We were introduced by a common investor in my nephrology company, Goldilocks Therapeutics, sort of got to know her. And about a year ago, she put together a business advisory board, and I'd been sort of advising her informally for a while, and she asked me to join. And my thought was, well, maybe I can get some stock in return for this. So that sounds like a great idea. With my nephrology company, Goldilocks, unfortunately, the technology, we couldn't get to work consistently. We returned it to Sloan Kettering. And when Stacy heard that I had some more time available, she kind of took me up on one of my suggestions, because for years I'd been saying, Stacy, you're awesome as a scientist, you're awesome as an entrepreneur, but you haven't done this before on the business side. Why don't you bring in an executive chairman? And so she called my bluff on that and said, Arthur, why don't you do that? And so for me, it's been wonderful. The first thing I had to do was call up a couple of my friends who've been executive chairmen and find out exactly what that entails, which is sort of anything. But it's been a great
experience. It's a mixed bag.
Well, it's kind of like being the grandfather, right? If you don't like what's going on, you go, here goes Stacy, deal with it. But I guess to me, one of the cool things, and I'll stop here because we have more interesting things to talk about, is through the years, what I was able to do as with my relationship with the company and on the business advisory board was to sit in on the SAB meetings. And, it's one thing to call an SAB member if you're a potential investor and kind of get the party line and all of that. But it's another thing to sit there and hear Neil Rosen and Andy Koff from Sloan Kettering bat around, not whether the technology is interesting, but sort of how to best go forward with this technology that they know is interesting to them. So, to me, that was wonderful diligence and kind of an easy call. So that's what brought me here.
Okay, so perfect segue. So Stacy, I want you to wind back a little bit, and you talk about how you're working on this protein as a target, and you always kind of thought it could be a potential drug target, right? And that's, I imagine in a lot of things with academia, there's a fair amount of affirmation bias, because you have things that you want to see, and like your brain wants to see that thing, right? So how did you kind of approach really the steps of saying, could this be a target, and what were some of the things that you did to kind of force yourself to really be difficult on it? Like how did you get it to a point where you knew and you knew you weren't fooling yourself?
Yeah, no, that's a great question. So I didn't start out thinking that I would start a company. I mean, I was a bench scientist, and then I was a professor, and I had just gotten tenure, and I envisioned I would stay and do sort of the same thing forever. But one of the first thing that actually happened is I submitted a really large R01 grant, which is the academic big powerhouse grant, and it got trashed. And, I didn't even get a score. And I called my program officer and I said, I'm sorry, this is a really great project. And he said to me, oh yeah, we agree. This is a great project. AIM 1 and 2 are fantastic, but in AIM 3, you want to make a drug. And you're an academic and you can't make a drug in academia. That's very naive of you. If you want to make a drug, start a company, get an SBIR grant, and make a drug. So that was the first idea. So I did actually drop that AIM from the grant. I repackaged the first 2 AIMs, and I got, a 5% in an era from the NCI where no one ever gets in at 5%. And I had gone from unscored to 5%. So they were being serious. They loved the idea. They just were like, you can't make a drug. And then at the same time, we patented this technology, and the head of tech transfer sort of took me out on a couple little dog and pony shows to meet venture capitalists. And naively, I thought, oh, they're going to love the idea. They're going to give me all this money. I'm going to go back into my laboratory and I'm just going to keep working. And suddenly they were like, well, no, if we like this, we're going to start a company and we're going to take it away from the university. And I said, well, why would you do that? I'm sort of the world's expert on this one little piece of the pie. I think that I should, help nurture this. So that was the second piece that said, if you want to do this, you should start a company. And then I think the third piece was I got really angry through this whole process. Like, wait a second, this is what I've been working on forever. And I think that I should probably continue to be involved with this. So almost on a whim, I applied to a program out of the New York EDC, which was the SBIR mentoring program. And I got this, right? They interviewed me and I suddenly had this force that could help me write this SBIR grant, which is very different than writing an R01. And I knew nothing about starting a company. So the next thing was I had to actually start this company, but I had two friends, one of who I'm married to, who is a serial, venture capitalist, a serial entrepreneur. And he said, yeah, we can start a company in 20 seconds. That's not hard. And I know how to raise money. The next other piece was I had a very good friend who ended up becoming my co-founder, who we had worked together on some boards, and she was not a scientist, but she was essentially a fractional COO. So she knew how to start companies. She knew how to get insurance and pay rent, pay electric bills, things that I didn't know anything about. I knew science. And so I suddenly had this company, and then I needed people to work in the company. And I had a really talented graduate student who was graduating, who had no interest to go to a postdoc. And he said, I'll come and work at your company. And then the last was we had an incubator at Downstate, where I'm a faculty member, the Downstate Biotechnology Incubator on site, 2 minutes from where my other office was. And I could get into the incubator. So suddenly I had this. So I think the point of the story was, as a scientist, we're sort of trained to see opportunities and see issues with data, to troubleshoot, to look at a panel of information and make patterns out of it. And I think that's what I did, right? There were all these things that sort of nature, that karma was throwing at me, that in retrospect were really saying you should do this. And I was able to sort of put them all together and string them together. And suddenly we had this company and we were off to the races. And I think that I had to learn everything about running a company, right? I didn't know what a cap table was. I didn't know what an addressable market was. But again, I'm a scientist, so I'm pretty quick study. I can learn things pretty rapidly. And I also understand that to learn things, you need to surround yourself with people that know those things. So that's what I did. I started going out and meeting people like Arthur, right? Who, we had some similar background. We had both gone to Princeton together, not at the same time, but, we had some commonality. And that was sort of the first kernel that I could say, hey, how about a coffee? And those issues were 100% like me learning. Every single person I talked to, I was like, just tell me everything. And I learned something from every single person I spoke to. And I think that was another thing that I was able to do because of my training as a scientist, was just, able to absorb this vast amount of information and was open to learning all these new things. So that's really the serendipity of it, but it wasn't really serendipity. I do really believe it was sort of the right thing, and everyone was sort of throwing these bits of information at me. And I was able to open my eyes and see this is the right thing to do. At the time, I will say, I did get pushback from a lot of people, both in academia as well as in business. And the pushback from academia was like, why do you think you can start a company? Why do you think you can, create a drug that could change the outcomes for cancer? And my response to that was, why don't you think that, right? For 30 years, we've been writing in our R01 grants. The first line of everyone's grant is, this could be a therapeutic target that could change the outcomes for cancer. And I felt very much a moral responsibility. I had been writing this in my grants because I believed it. And if I really believed it, then I should really do something with it. And so I was very critical of a lot of my colleagues who said, why do you need to do this? You're tenured, just, sit, you've got a job for the rest of your life, just do your work. But I felt that I wanted more because this was what I had been thinking about for 30 years. And I also did get pushback from business, from biotech, which was sort of what Arthur was alluding to. A lot of people said, why do you think you can do this? You're a scientist. What do you know about business? And my comment to that was, I can learn about business, but I know science, right? And I know what science has taught me is how to cherry pick what I need to know, how to build great teams, how to mentor people, how to assemble a business plan and a work plan and to see the path forward. And what I bring to the table is this deep understanding of the target in a way that I think no one else really had thought about this target or this pathway, and to have the vision, because that's what I do. You do as a scientist, right? You think deeply about something and look to the future. So my comment to the business world was, yeah, I can learn all the business because you're going to teach me, because I'm going to interface with you. And I'm going to bring something unique, which is this ability to understand this mechanism really better than anyone else. And I think I would like to believe that, 5 years, 6 years out, it's served us well, right? We actually are, a company, a really strong team, and, are really poised to bring a transformative target to the market.
So, Stacy, how long between the time you got mad and standing up Concarlo? How long did you work the problem in order to end up with an actual company?
I didn't work it very long. So I probably got the reviews from the grant, the summer of like 2016 or something like that. Or I probably got them in the early summer, and I festered for like 2 or 3 months. And it was the same time in 2016 that I had gone to, some of these dog and pony shows. So it was honestly like an email that came into my inbox from the New York EDC, and this conversation that I finally had with my program officer that got me really mad. And, it sort of felt like, wait a second, it's so hard to get R01s anyway. You're telling me I have a great idea, but I'm reaching too high with this idea. And that's what made me mad. And so it was really a whim. And it was also the support of all these other people. It was my husband saying, looking, I was showing him some data. We had this beautiful data where we were getting this massive regression in this mouse model. And he said, wait, I don't know anything about cancer, but don't you want the tumor to disappear? And you've made this tumor disappear. And I said, yeah, of course, it works really well. And he said, I don't know, that seems like that you should do something with that. So it was tapping into my personal network of people believing in me and saying, yeah, you got this, right? You can do this. And, reaching out to different people saying, yeah, this sounds like a good idea. And also being willing as a scientist to try things, because that's what we're supposed to do. We're supposed to take a hypothesis, we're supposed to think deeply about it, and then we're supposed to try. And you're supposed to design the experiment so it's not a binary, like it failed or it worked. If it doesn't work, you're supposed to understand why it doesn't work. So we decided to start the company that way. And, back to Derek's question, I spent the next 3 years at the company trying to prove myself wrong. Honestly, really trying to set up experiments that would show me that I was wrong, because I was probably the most critical of everyone. And, I never used the words. If you go back and listen to me speaking, I was much more cautious in my language back then. In terms of talking about the power of the target and the transformative potential, because I was doing my own diligence, right? I was creating a scenario where I was like, well, let's do this really hard experiment because I think it probably won't work. And then it would work, or it wouldn't work exactly the way we thought it would, but it would show us a path forward. And so I think, again, bringing that deep scientific process to this early work was crucial, and being very data-driven and being very willing to look at the data and say, there is no good data or bad data. There's just data that's going to tell us what to do next. And, I think it happened quite quickly. It was probably the summer of 2016, and I had this company by, the winter of 2016, and then was starting to talk to people and did have moments of like, what did I get myself into? But we signed we moved into the biotechnology incubator in January 1st, 2017. Okay. And suddenly, we hired people and we were doing experiments and buying equipment. And I was like, well, let's just see how this goes.
Yeah.
I think we've all had those moments of various things in our lives. What do we do? And most of the time if we keep going, then they yield results.
Yeah. All right, so Arthur, I'm going to tee you up on this one because so I absolutely love the positioning of the company, which is cleaning up what precision oncology leaves behind, right? And I love this because we a lot of times fall into a trap and saying, well, there's an effective therapy for this part of cancer? Why do you want to pursue something like that? And I love the fact that what you're doing with this target and with the company is like, no, there's a lane that fits in. This is where it works, and this is how it fits into the entire kind of overarching realm of cancer care. So you could talk a little bit about that with Concarlo, but you've also seen good biotech stories and ones that kind of don't quite hit. So what is it about a biotech story that really kind of makes it hit for you, both as an investor and now as an entrepreneur?
Yeah, good question, Derek. And I like the use of the word story, because I do look at these companies, especially when you're out there marketing to investors, as a story. And listening to Stacy talk about kind of the transition and the growth beyond being just a scientist and having that other arrow in her quiver, there's a real difference between the slides that a scientist will show. And I laugh because I tell groups that I mentor that if a VC is just going to read the titles of your slide, just flip through each slide and read the title, they should get your story. And so your technical slide, if there's just one, should say, positive mouse data show blah, blah, blah for your VCs, whereas the scientist won't want to tell the viewer what the conclusion is. They'll say, here's the mouse data, right? And not insult the viewer with a conclusion. So I think it's a very different approach. And so, yeah, if you think about then what kind of stories make sense, they need to be understandable. And Stacy is, as you might have been able to tell, a very terrific communicator. The story, the molecular biology is not so simple, but the conclusion really is. And we all know that cancer is not cured. We've made great strides, but anyone who's had a friend or family member diagnosed with cancer, you realize how far we have to go. And this is just one of the pieces. So it builds on something that is really kind of the latest wave, precision oncology, which is wonderful, but it's not the be all and end all.
And it's not perfect, right?
Exactly.
We fall into this trap of like, well, I thought the BRAF inhibitors were really good. It's like, well, they are really good, but they're not 100% really good. And nothing it's precision, but it's not perfect. And we as an industry, we're like, oh, this is the overall response rate. And if that overall response rate is less than 100%, that means it's not going to work for everybody, right? So we get caught up in therapies that are actually quite good, but we assume that, oh, if you have cancer type X and there's a therapy that's good, you're in the good group, right? We often don't really think about the fact that it's kind of not perfect, and there are these patients that aren't necessarily well served, even by the therapies that are heralded as great, which they are, right? But it gets much more complicated than that when you get into, this is the way it works in this binary patient. When that one patient is the one that you really care about, that's the one that's the most important to you.
Yeah, absolutely. You see these heralded approaches, and Stacy talks about it, and then you look at the data, and sure, it's a statistically significant and important 3 or 4 month gain in lifespan, which is awesome. But there's a whole lot of room for improvement, and ultimately, multiple approaches are going to be required. And I'm enthusiastic about what Concarlo is doing in particular.
Mm-hmm.
Yeah, maybe I could add to that as well, which is I think that you hit the nail on the head, Derek. Right? We frequently think about this in two siloed. I'm going to create a drug against protein X that's going to do Y, forgetting that everything in the cell, but everything in particularly in the pathways that drive the proliferation of cancer cells is a pathway. And so you have to take a step back, in my opinion, and look at that pathway in toto, right? From the cell surface all the way to the nucleus, because you're putting one piece of gum in the system, but there's lots of room for evolution away from that blockade. And there's lots of other modalities, other targets that can get into that to overcome what we've done. And it's only when we start thinking about it as a continuum, and I use this phrase, the oncogenic funnel, which is really the proliferative signals, the signals that are driving a cancer cell to divide, that go from the cell surface through the cytoplasm, so the RASs, the RAFs that you talked about, all the way to the nucleus. And until you think about it as a continuum, there's always going to be ways for that highly evolving tumor cell to get away from the blockade you put on. And that's really the problem, right? Those RAF inhibitors you talked about, they're really good. They're going to hit 99% of the tumor cells in the patient that's driven by that RAF mutation. But there's that 1% that is going to evolve away. And that is the 1% that is going to come back and cause that drug-resistant phenotype. And so we need to be thinking about that 1%, even when we use that first BRAF inhibitor, because that 1% is a lot of cells, because remember, tumors are not, 100 cells, they're 1 times 10 to the 9th cells, or 1 times 10 to the 13th cells. And 1% of 1 times 10 to the 9th is actually significant tumor burden that is rapidly evolving away from the blockade I just put on. So what we do at Concarlo, and what we talk about when we talk about leaving what precision oncology leaves behind, is a tribute to precision oncology. It's the understanding that we know so many nodes in that oncogenic funnel to target, but by targeting just 1 or 2 modes independently, we're leaving a lot of room on the table for that evolution to occur. And so we want to target at the bottom of the funnel, right, which is where our drug IPY and P27 sits, with the idea that, going back to my funnel analogy, it's a lot easier to block the flow of water by pinching off the really narrow bottom of the funnel than to block the flow of water at the top of the funnel or the middle of the funnel. There's always ways for that pernicious water to get around the blockade. If you pinch off the bottom, you're going to have a better outcome. And so that's what I think the field is coming to, with this desire to drug effectively CDK4, CDK6, and CDK2. And then we're going to add an extra layer on, which is it's not okay to drug them independently. You've got to drug them simultaneously, because they too operate as a seesaw or a continuum. And what you do to one group of proteins is going to be felt by the others. And I think that is where the evolution of our field is moving, and where we really sit, is thinking about all of this pathway together. And so that's what I think we really mean by saying this. And this is a way to help those frontline therapies work better, as well as to deal with patients that are now have been on 4, 5, 6 prior lines of therapy, by really going to the bottom of that oncogenic funnel.
As a non-scientist, I'm glad that I looked at your graphic of your funnel on your website before, and I'm actually going to put it in. I'm going to put that in the chat for everybody.
Oh, really good.
Great.
Yeah, so that they can take a look, because I think what you described was visual, but I think the way you have it set up on your website also makes it even more visual. And
we look at a lot of these, and a word of appreciation for making it easy, good, and simple to understand and digest. It really it's a lot of time, but it's excellent. It's so nice when you look at it and you just get it.
Oh, well, thank you. I mean, again, I think going back to my training in retroviruses and HIV and Maloney-Marine leukemia virus, right? The success we've had in many other diseases, such as HIV, is exactly we're targeting multiple nodes in the pathway, recognizing that a virus, very similar to cancer, is a highly evolving entity. And it's not okay just to hit one blockade, because that virus or that cancer cell is going to very quickly outsmart the blockade that you put on. And the success that we've had in other fields has really showed you've got to cripple it in multiple nodes, because yes, 90% of it is going to be crippled with, 99% of it probably is going to be crippled with the first node. But what you need to deal with is that 1% or that 0.1%, which is going to come back and cause the problems for our patients. And so I do very strongly agree with Arthur. We've had amazing drugs that have come to market, but we need to actually keep pushing, because it's not okay just to extend progression free survival by 3 or 4 months, or overall survival by a few months. We need to make now these bigger gains, and to really leverage our understanding of these pathways to, be smarter, and to create better drugs that will deal with that pathway in toto.
So let's switch gears a little bit. We've had a question from the audience about some of your investors. So let's talk a little bit about, you got mad, you in a very rapid succession did the steps to stand up the company. Part of that obviously has to be fundraising. And so the question from the audience was, how important was the support of investors like the Catalytic Impact Foundation and Princeton Alumni Angels been on your amazing journey? So let's just talk a little bit about sort of your fundraising journey and building a company in this case, right, in New York, in the city.
Yeah. So, so we started the company and then we needed to raise money. We did apply for the SBIR grant. We didn't get it the first time around. We did eventually get it, but, we had to raise money in the meantime. And so we went out to sort of high net worth individuals, friends and family, and we sort of raised money from that pool originally, people that knew me, knew the technology a little bit. And then we started going out to get some external validation. And that's where, the Catalytic Impact Foundation and Princeton Alumni Angels, we got money from New York State, the Accelerator Fund. That was really important because that was really external validation of the technology of the team. And that was crucial at that point. It was crucial to help with additional fundraising, but also it was really important, I think, for me and for the team to, again, I was trying to knock myself down, right? And I wanted other people to say, sorry, I'm like choking here, other people to say, yeah, this is a really good idea. So it was really important that we got that funding right away. Raising funding is hard, and I feel like we've been doing it pretty much nonstop since we started, which I guess is what you have to do. And we raised a couple different seed rounds, and then we did issue safe notes. And now we're really in our first big institutional raise. We're raising our Series A round, and, bringing Arthur on board in the fall was really essential to that effort, to go out and to really demonstrate we have an even stronger team. And, just getting out there, talking to a lot of people, telling them the story, trying to get more people to believe in the story. I think that's the bigger thing that we spend a lot of time doing. Arthur, maybe you want to have some comments too?
Yeah, I think the venture capital funding world has changed, certainly since I've been involved in the industry. And as you all know, a lot of VCs are doing company formation, which I think is a great thing for them. They kind of own the whole technology, the whole company. So it would certainly lead to higher returns. So nothing against that as an approach. But what that means for a startup is you're now not just competing with other startups. You're competing with what the VC is doing themselves, and effectively is going to own the whole piece of. So that makes it harder, and I think that has increased the importance of groups that the foundations, Catalytic Impact, Angels, etc., the groups that will help take a company like Concarlo from the pure startup mode, where you're competing with those, to the next step, where now if a VC is looking at Concarlo versus something they're incubating themselves, they're kind of in different buckets, right? And they can do both if they want to, but they don't have to sort of choose one over the other. And that's what we're hoping to do now as we get out to raise a substantial Series A round designed to take the initial product not just into the clinic, but through for phase 2A efficacy results.
The other thing I'd say about fundraising is that, when money is tight, you have to run a lean, mean ship, right? Which is what we've done, right? And so everyone, I give tremendous credit to the team. Everyone, works all the time and does many jobs. We all do, from I feel like I'm like terrible at, sending out FedExes, but I do that frequently, right? So, we all have to do more than what we would do if we were a flush company. But I also think that makes you really evaluate every experiment you're doing, every dollar you're spending. And I think that is a really important thing, right? So that, is this adding value to our product? Is this adding value to our story? Is this adding value to our message? And if it doesn't meet those three criteria, maybe we shouldn't be doing it right now. And that is, that's important, right? There's only so much money in biotech in general. And so we want to be good stewards of the money that we have raised.
Stacy, there's a conversation that hopefully we'll have a chance to have after we raise the big Series A. But when I was on the VC side and we would make an investment and join the board, sort of by definition, those companies were now sitting on a whole lot of cash that they didn't have before. And to me, the first thing you had to figure out as a new board member was that CEO who was used to being cash poor, was he or she then going to spend the money too freely? Oh, I've got all this cash. We're gonna buy a new facility. Or where they're not going to spend it enough. I've been poor before, and I'll never be poor again. And literally, you would decide as a board member, is my role to tell the CEO to spend or to stop spending? And again, Stacy, we'll try to figure that out, but it's going to be interesting, and I hope we have that opportunity.
Yeah, no, definitely. And again, I think that, I have run a lab for 20 years. And so that lab has come from federal money or grant money with very strict budgets, right? And when that money runs out, you're done working. So, we've definitely, I've learned how to budget. And so I was thinking, someone was asking me, well, how did you come up with this? We're raising 55 million in our Series A round. How did you come up with that number? And you're like, well, that's the amount of money that we need to do this work, right? We budgeted it, and it's a very clear, we have clear milestones, and this money is going to do certain things. So there'll be no purchasing of gold faucets for the bathrooms or anything like that, because that's not in our budget. And so I think that seems very obvious to me, but I guess that is a pitfall that frequently CEOs fall into, and that will not happen at Concarlo, I can tell you that.
No $100,000 shower curtain.
No, exactly.
Well,
so.
it's also like you alluded to, Arthur. It's the false promise of runway, right? Where you're like, we have enough runway for 4 years. When your venture capital is like, yeah, well, I want to sell it in 3, so how about you spend it in 2, type of thing, right? So when you're thinking about a Series A, right, you want to get, you have a target that you want to get through, whether it's concept, whether it's phase 1, phase 2, etc. So how do you approach it to think about what's the right size chunk of money to ask for? And, how does that discussion usually go if there are, do you have staged plans of like, okay, this is what we could do with 50, and this is what we can do with 10? And all of this is really rich, considering Arthur's, domain's original fund was $41 million, right? And that was the entire fund. So, maybe you could kind of both take a step at this, because I would be very interested to hear how you're thinking as a company. And Arthur, I would really like to hear kind of the venture capital point too, which is like, well, we see a company that asks for, 50, did they ask for too much? Are they not asking for enough? Right. So it'd be great to get two sides of that coin.
Yeah, so I think you start with what are the key value driving goals that you're going to achieve, right? And how, because the VC really is going to be looking at how are we going to raise the next money at a substantially higher price. So for Concarlo, we believe that having phase 2A data showing not just safety, but some signs of efficacy are really what's going to drive value. And so, then we sharpen our pencils, get to a number to get there, which, as Stacy mentioned, we believe is $55 million. Mm-hmm. That doesn't mean that the VC syndicate that hopefully will come together soon is going to sign on for that and write a check for $55 million on day one. They'd rather perhaps keep some of that money in their pockets and have the company achieve some interim milestones. But I think that that's really what the key is. And the other thing that I would just point out on valuation, entrepreneurs tend to focus a whole lot on pre-money value, right? Which sort of makes sense because that's sort of the beginning of their dilution. Whereas the VCs focus more on post-money value. So however we get there, if we spend the $55 million, the question that they're going to ask is, well, what's the value of the company then compared to the post-money value that I invested in, because that's my step up. And I think that's a real disconnect. And, kind of as I think about my background on both sides of the table, and I do a bunch of mentoring, I do try to translate between the two sides, the two different mindsets.
Yeah, I would echo that and say that when we sat down and tried to think what would be, what would really transform the company, the value lift. And the value lift is, demonstrating that this target, which I think everyone in the field agrees is a high value target, but hasn't been drugged directly before, that drugging it is tolerable in humans, because remember, humans are not big dogs or big mice. And so it can be really tolerable in those species, but we have to actually show it's tolerable in humans, and actually design the trial that, 1B, 2A to have show efficacy, and not just looking only for PR or CR, treat it like a science experiment and have really good pharmacodynamic markers so that we can show that the drug got to the tumor, that the drug engaged with target, and that the drug actually caused outcome, whether that outcome was sufficient at that concentration to cause that PR or CR, that's a different story. And so design that trial so that we walk away with really showing efficacy, showing tolerability. And that was what we said is going to really transform and sort of crack open the concept of drugging P27. And so, and that trial is expensive, right? You know, to run clinical trials in oncology it's probably, we have $25 million budgeted to run that trial. And so as Arthur was saying, you know, we'll get to the IND and that would be great. And that's going to be amazing, but that's not going to really transform this space. And so that's why we structured the raise to really get us through that 2A milestone. We hear frequently like, oh, I believe this target is really interesting. It sits in a very well clinically validated path. We actually have sort of like clinical validation that the target's important, but no one's actually ever drugged it. And so show me some human data. And that will be, I think, the calling card that will change the story. And so we also recognize that and have factored into this fundraising R&D money, because we have other ways that we're drugging the target besides our lead, and we want to really have that space flanked. By the time we demonstrate the value of this target and where it sits in this pathway, we want to be able to then have our secondary and our tertiary targets moving through the pipeline so that we're ready for different tumor types, different disease indications, really recognizing the value of targeting something that sits so far down in that oncogenic funnel.
Yeah, you want to kind of take it as far away from a kind of single molecule binary event as you can and say, okay, let's get multiple bites at the apple because we know this is going to be clinically important. How do we stay out of kind of the dreaded gray area, and how do we make sure we can get to a point where we can actually show, you know, real efficacious data?
Exactly.
So we had a question from the audience, and you've actually answered several parts of it already, which is good. But one piece of the question that is still out there is, have you engaged pharma yet? And have you started talking with pharma companies that are in the oncology space?
Yeah, we've started that. You know, we've had some good conversations with both venture arms of some pharma as well as the BD arms of certain pharma, both pharma in this country as well as pharma from other countries. I think that the concept of drugging CDK4, 6 and 2 is really first and foremost. And I would point out that these targets, which really drive the proliferation of all cells and all tumor cells, has really been a goal of the oncology field since its inception over 40 years, right? We've been trying to effectively drug these targets, and it's gone through fits and spurts. You know, there was a big push in the 80s to try to drug these targets, and it didn't really work. And then it petered away, and then, you know, it sort of came back with the Ibrances and Verzenio's of the world, Kisqual. And, you know, those are great drugs, but now we see the limitations of those drugs, right? That essentially almost all patients on those drugs will become resistant to those drugs with time. And that's anywhere from 3 months to 3 years, but that evolution is going to happen. And we need to have now secondary therapies for those patients. And so that brings us back to CDK2. And so there's a lot of interest to drug CDK2. And that's sort of where pharma, I think, perks their ears up when they're like, wait, you're trying to drug all 3 of them simultaneously. And then, you know, P27 is, I think most people would agree, you know, they know where it sits. It interacts with these 3 kinases. And as Arthur started out, our scientific advisory board, real leaders in this field, they are 100% on this target. And so you would say, well, why haven't we drugged it before? Because it's difficult, right? I mean, it's not, we as a field, and I use that loosely, you know, we are really good at making monoclonal antibodies, so we can drug the top of the cell, and we're pretty good at making small molecule kinase inhibitors, so we can drug all the kinases, but we're not as adept at drugging things that don't fit into those 2 classes. And so this is a intracellular nuclear target that doesn't have an obvious pocket. And so you have to be creative and of how to drug it. And so I think that is where we sit in, and talking to pharma, I think they're recognizing the value of it. But again, there's that, you know, hesitation. Well, it's never been drugged before. And that brings us back to, where did we come up with that 55 number, which is, we're going to show you that what happens when you drug this target and that it's tolerable and that it's efficacious, and that this is a brand new way of dealing with the resistance, not only seen in the presence of Ibrance, but also in the presence of those RAF inhibitors that you talked about, or Herceptin, or Tarceva, any of those drugs. And so that will then expand our world of possibility to drug it by lots of different other modalities and in lots of different tumor types. So, I think we're getting some engagement with pharma, but again, those are ongoing conversations.
It's funny. As soon as you said it was hard to drug, I immediately went in my brain to, well, have you tried artificial intelligence, right? So, so in thinking, but the real question is, there are a lot of different platforms now for trying to make drugs smart. We've actually done pretty well with structure-based drug design. So how did you initially start with, what was your discovery funnel like, and how did you get to the point where you had, candidates that you like? Because honestly, that part of the field seems to change every 10 papers that come out. This better. I'm pretty sure if you just type in what you said.
Hey, Derek, you
Derek,
lost your audio.
All
right, am I back now?
You're
you're
back.
back.
You're back. I think I got half the question, but
All right, you can see where this is going, right? That part of the field seems to change every 5 minutes. I had a really good joke about, just typing it into Chat GPT and seeing what molecules that came up with.
yeah.
So, but honestly, how do you think about that in terms of the way that you get your candidate molecules? And, is it something that's even possible to stay on top of, or do you just kind of have to be diligent and say, these are the things we want out of a molecule, and we have the ability and the connections to go get them?
Yeah, I think that we went back to the biology. We went back to the mechanism. And I always say, P27 is the natural inhibitor of CDK4, 6, and 2. That is the way the cell normally inhibits these 3 high-profile kinases. And you know what? Nature's always going to be smarter than we are. So I'm going to try to get P27 to do its job. And our lead compound is actually a peptide, and it's actually based on the natural inhibitor of P27. So P27 inhibits these 3 kinases. There's another protein in the cell that actually inhibits P27. And we said, nature's already figured out how to do what we want to do. Why wouldn't we just use that? So we actually took that peptide and we bioengineered it to make it smaller and to make it more stable, to make it more drug-like. And then we actually package it in a liposome. So similar to, the liposomes that we all have in our arms right now and from the COVID vaccines. And we can show that increases its serum residence time, and we can actually get delivery to the tumor, and we can turn off the target within, an hour, and we can get the tumor cell to start dying within 24 hours. So that was our lead, was really going back to the biology. And again, thinking like a cell biologist, which is, I understand how these proteins move around. I'm going to leverage that. And then we now do have other modalities. We have other assets in our pipeline, including small molecules against the P27 target. And we did the same thing. We said, let's use nature that has certain complexes. Let's design assays that mimic those interactions and create functional assays, which is what we did. And that's how we interrogated our library. So we didn't just say, we're going to look for, here's our target and here's the small library, and we're just going to see what binds. That didn't seem effective to me. And it also wouldn't really work for a protein like P27, because P27, like a lot of proteins, doesn't have structure on its own. It only has structure when it binds onto its targets. So we said, let's interrogate a library and have the library do things that we know P27 does in the context of its in vivo situation, how it interacts, who's it talking to, where is it located? Let's make the screens that way. So we took a sort of, I'd say, a science-based functional approach. There is some AI that's brought into that. But again, back to, you got to understand the biology. You've got to create the assays that mimic the biology to ask the right questions, or you're going to use a lot of these fancy techniques to come up with something that doesn't actually translate into dealing with that complex biological situation. And I guess that comes back to what we were talking about before. You got to think about this as a continuum, an entire path from the cell surface to the nucleus. And I've got to think about all the things that these proteins do. Where do they move? How are they modified? How are they changed? And design therapies that fit into that and leverage that. And so I think that's sort of our sweet spot, is understanding this biology probably better than anyone, and then using that knowledge to harness proteins that are already in the cell, and also using that knowledge to come up with new entities, new composition of matter based on that knowledge as well.
I think Concarlo is an interesting story because we're not a traditional platform company. It's not mRNA or something like that, but we're also not a traditional product company, a one-trick pony. And so the whole idea is to really own this P27 space. And we believe that's going to be a really important target. As Stacy said, the initial product is a peptide. If you're going to interact with a protein, it's easier to do that effectively with a bigger molecule. So that's good, but peptides come with their own problems. Small molecules certainly easier to manufacture and put into people's arms, but asking a small molecule to do what you want it to do on a floppy protein like P27 is a big ask. We think we have some real leads toward getting there, but the key for us is we want to own P27. And that means both offensively figuring out what works, and defensively figuring out how to stake out the landscape so that we can really own the protein.
Can we let's shift? I feel like I've been to science class, which is amazing. This lawyer has learned some science today. So let's talk about, we have only a few minutes left. Let's talk about building the company in New York. And Arthur, let me start with you, because this is not the first company you've helped to build in New York. So talk about standing up a team, scientists, other types of professionals that work in life sciences in New York, and that process.
And you can contrast now from like 8 or 9 years ago.
Yeah.
Well, I was thinking about that, and I don't know if it was on this program or a different one, but I saw Mark Tessier-Levine when he was leaving Rockefeller after having come from Genentech to head up Rockefeller and then to go be president of Stanford. He was sort of giving what was supposed to be an inspiring New York biotech speech. And he said, I believe that in 10 years, New York can be where Boston was 5 years ago. And to me, that was just such the wrong comparison, because I think New York is very different from Boston, and I'm not just talking about the sports teams.
Yeah.
But everybody knows we've got the academic institutions. Everybody knows we've got real estate spaces, issues that have been tried to address, and to some extent have been addressed. But I think what's really started to happen over the last bunch of years is sort of critical mass on the part of the investigators at these world-renowned institutions. And one thing that Stacy didn't mention, and I don't know what happened when she formed the company, were there a couple of people down the hall who had done this before at SUNY Downstate, and I'm guessing maybe not so much. Whereas I'm also guessing that as she's been at this for 5 or 6 years, a bunch of people have come to her and say, hey, how'd you do this? How's it working out? I do a lot of stuff with Sloan Kettering, and they are perhaps more advanced at this, but it's really been largely over the last 10 years where, just about every professor has their own startup or two, just like at Stanford and Harvard. And so I think New York has come a long way in terms of the critical mass at the institutions, bolstered by the resources we came with and the ones that EDC and others have provided elsewhere.
Yeah, I would echo that. I felt when I started, I didn't know anyone that had done this, and actually really had a hard time finding anyone who had done what I was doing. But now in the last few years, I feel that there's been an explosion, a good explosion of programs and opportunities to learn about how to do this, and mentorship programs and things like that. I wish that I had had access to a lot of those when I started, but that just forced me to go out and find the people on my own. And you can probably tell I like to talk and I'm not shy. So did go out and dig those contacts up. I did participate in, like the Springboard Women's Entrepreneurship Program, which was really great. And that was, if nothing else, a great opportunity to meet other women who were becoming CEOs, who were actually starting companies, because I hadn't really met any of them. There is still very, a gender issue in terms of biotech. And so it was nice to meet other women that had come from academia, who had done this and who were running their companies. But I think that it's a lot easier now. There are more resources, and I think that's a great testament to New York, the ecosystem. One last thing I would say is, it is still difficult to hire people in New York because we don't have a lot of the sort of the concentration of biotechs that exists like in Boston or San Diego. So whereas one biotech shuts down, there's another one nearby that everyone can just flop on over to. We don't have that. So we do have a harder time getting people in, which has forced us, again, back to the Zoom society. Some of our team is remote. We have people that work in Vancouver, we have people that work in San Diego, and it works just fine. And so I think that is a good thing about the pandemic, that it is actually made normalize that ability of people working all over the place and just coming in and out and doing work via remote. And that, I think, is something that's going to continue to help New York City, because we, to get people who have that drug development experience into our teams in New York is going to be more difficult, because we don't have as many, biotechs for people just to hop laterally to.
Hopefully we will be getting there, right?
Yes,
As companies like Concarlo and others grow and expand. Stacy and Arthur, thank you so much for joining us. What an inspiring conversation we had today. Everyone, I hope you have a great week. We'll see you next Tuesday. And don't forget to register for the Bio CEO Welcome Reception on Sunday the 5th. Everybody, thanks so much.
Thanks so much.